dengue is a great topic to talk about diagnostics because it actually, the principle, if you understand the principle, actually applies to quite a number of zoonotic diseases or vector-borne transmitted diseases. And the principle is that once you get bitten by a mosquito, the virus goes in the skin, is taken up by skin cells. These skin cells, loaded with the virus, go to your lymph nodes, wherever they are, and then there is a replication phase, and then they spill over into the blood. And then you have a lot of virus in the blood, like really a lot, like millions. And this is called viremia, the virus in the blood phase. And this phase sort of, comes up and then it comes down again as the human produces antibodies, because the antibodies then bind to the virus, and the antibodies are made for our specific macrophages, so cells, monocytes, macrophages, to take up opsonized, so proteins which are coated in these antibodies, and then they're taken out of the blood and they go into tissues. So you have a phase in the blood, and then you have an antibody phase. And this is exactly how you should design your diagnostic strategy. So in an early stage of the disease, you would look for proteins of viruses in the blood or for DNA of viruses, whereas in the later stage, you would look at antibodies because they then come up. So for dengue, there is a double test. There is a NS1, so it's an antigen. It's a non-structural protein of the virus, NS, non-structural, which is picked out by an antibody. And then in the second phase, you look at the IgMs or IgG antibodies, and taking those together, you cover the entire spectrum of the dissemination and the immune response, and you're able to diagnose with very high accuracy dengue.